Good afternoon, everybody. Welcome back from lunch. I'm Tom Ciulla. I spent nearly the last 30 years in clinical research and in academic and industry leadership positions and worked on multiple drugs that have achieved FDA approval. As a chief medical officer at Clearside Bio, I supported the first FDA approval of a suprachoroidal therapy, as well as partnerships with REGENXBIO and Aura Biosciences, who have a gene therapy and a virus-like drug conjugate in clinical trials using suprachoroidal delivery. I'm very excited today to talk about suprachoroidal drug delivery. We have a fabulous spotlight on suprachoroidal drug delivery today. After my overview, we'll have some excellent company presentations, and then an expert panel discussion co-moderated by myself and Emmett Cunningham. Let's talk about suprachoroidal drug delivery in practice and clinical research. Suprachoroidal drug delivery can be achieved via catheter-based and tissue separator-based systems, as well as microneedle-based systems. And we'll hear a lot more about that today during the company presentations. The first FDA approval of a suprachoroidal therapy, XIPERE, in 2021 has established the foundation for in-office non-surgical delivery. This videotape shows the administration at the pars plana, and you can see how the injectate spreads circumferentially and posteriorly. There's been rapid expansion of suprachoroidal drug delivery in clinical trials, and we'll hear more about this today during the company presentations. Let's talk about the biomechanics of suprachoroidal drug delivery. What are the core benefits of treating via the suprachoroidal space? Well, it includes the ability to target chorioretinal tissues for potential efficacy benefits, the ability to compartmentalize drug away from the front of the eye for potential safety benefits, and bioavailability. After all, the chorioretinal tissues are essentially bathed with drug after suprachoroidal injection. There's circumferential and posterior spread of injectate after microneedle-based suprachoroidal injection. These images show a suprachoroidal injection of a fluorescein dye in ex vivo porcine eyes. And you can see on the left how the injectate spreads circumferentially from the injection site to the posterior tissues. On the right, you can see how the injectate spreads from the scleral spur posteriorly towards the macula. What drives the injectate posteriorly? This is a very important primate study where the hydrostatic pressure of the suprachoroidal space was measured. In the upper left image, you can see this pressure-sensing catheter behind the macula in a monkey. And on the table on the lower left, you can see the relationships where the intraocular pressure is greater than the anterior suprachoroidal space pressure, which in turn is greater than the posterior suprachoroidal space pressure. On the right, you can see that these pressure gradients persist regardless of the intraocular pressure. So importantly, these gradients drive uveoscleral outflow, and they maintain the suprachoroidal space as a potential space. Importantly for us, these pressure gradients drive the injectate posteriorly. These OCT images, all of which we've published, show expansion of the suprachoroidal space post-injection. In the top image, in a rabbit model, you can see suprachoroidal space expansion posteriorly all the way to the optic nerve head. In the middle image, you can see in a clinical trial subject, the suprachoroidal space expansion immediately anteriorly after injection. And on the lowest image in a clinical trial subject, you can see suprachoroidal space injection posteriorly. Let's talk about the versatility of suprachoroidal drug delivery with respect to small molecules, gene therapy, and virus-like drug conjugates. There's durability in the suprachoroidal space for particles ranging from the size of small molecule suspensions to DNA nanoparticles to AAV. So this is a very important study where various particles were injected into the suprachoroidal space in this rabbit model, and you can see from the graph on the left that all of these particulate suspensions last for 40 days or longer in the suprachoroidal space. And that's most likely due to the small choriocapillaris pore size, which measures about 6 nanometers, so much smaller than any of these particles. Preclinical data supports the durability potential of small molecule suspensions delivered into the suprachoroidal space. So this is data that we've published previously, and you can see these various small molecule suspensions. So triamcinolone, the active ingredient in XIPERE, a complement inhibitor, a plasma kallikrein inhibitor, and axitinib all last to the length of this experiment, out to 90 days. And these exposure levels are several log orders greater than the relevant IC50. Gene therapy is classically delivered via subretinal administration. You can see in this Luxturna patient that the subretinal injection results in a localized dome-shaped temporary detachment of the retina that we call a bleb. However, the direction and expansion of the bleb is not predictable. So in this particular case, you can see the perfluorooctane bubble trying to protect the macula. We know that there's risks of macular hole, as well as risks of hemorrhage from the retinotomy and fibrosis from the retinotomy. So therefore, that's led to an effort to develop viral vectors for alternative routes of administration that are less invasive. We're going to concentrate on AAV8, which REGENXBIO is administering suprachoroidally in their current clinical trials. This is another very important study looking at retinal cell tropism after suprachoroidal, subretinal, and intravitreal injection of AAV8 in non-human primates. In the top image after suprachoroidal injection, you can see this widespread expression of GFP. In the middle image after subretinal injection, there's focal expression. And in the upper right image after intravitreal injection, there's very little expression. Suprachoroidal delivery of RGX-14 produces therapeutic levels of an anti-VEGF Fab in rats. This is work out of Peter Campochiaro's lab, and you can see on the left when this is administered suprachoroidally and subretinally, the levels are essentially the same. REGENXBIO is conducting 2 trials, AV8 in neovascular AMD and Altitude in diabetic retinopathy, assessing suprachoroidal RGX-14, and Steve Piccola will talk much more about this later. I want to briefly talk about virus-like drug conjugates. Belsar is a virus-like drug conjugate with a novel dual mechanism of action. It's delivered suprachoroidally to achieve very high levels at the choroidal melanoma, which is targeted and bound. It has 2 mechanisms of action, including tumor cell necrosis and immune-mediated tumor killing. One last slide on Belsar. This is a rabbit model, which includes a human choroidal melanoma. You can see that tumor on the left. And when this eye is treated with Belsar plus laser radiation, you can see how the tumor essentially melts. So I've covered a lot of ground on suprachoroidal drug delivery, including the core features of suprachoroidal injection. That is the ability to target chorioretinal tissues for potential efficacy, to compartmentalize drug away from the front of the eye for potential safety. We talked about how the injectate reaches the posterior pole via these pressure gradients, and I've just briefly highlighted some of the versatility of this administration procedure with respect to small molecule suspensions, gene therapies, and virus-like drug conjugates. So thank you for your attention.